Okay, so we're starting the first chapter for our last lecture unit, and there's two goals of this lecture today. The first goal is that you understand the structure of DNA and the history behind it. That's because you're going to be tested on those scientists and the structure. The second takeaway is the process of DNA replication. So as I finish each one, I'll say, okay, like check, we've accomplished what we needed to for this part. So we're going to begin with DNA structure. And as with most of my lessons, I want you to understand where we are or how we got there. So to begin with, do you remember what DNA is an acronym for? Deoxyribonucleic acid. One of our very first lectures, I said, I just want you to say it like in a group of friends. You just sound smart. You're like, deoxyribonucleic acid. But why do you think we need to dedicate an entire chapter to deoxyribonucleic acid in biology? That's what makes us unique. Keep going. Everybody's made of it. Close. Like you're getting there. What'd you say? It is a part of life. What do you mean it's a part of life? It's a part of you, but are we the only things alive? So help me here. Plants, okay, it's a part of plants, it's a part of us. Keep going. What did you say? Any living organism will have DNA, and guess what biology is? The study of life, which is why we can justify using an entire chapter on DNA. Now, the second part of this slide that I want to point out is this picture right here was the cover of Time magazine in 1953. This is James Watson and Francis Crick. This is right after they discovered, and I'll come back to this in my history lesson, of course, they discovered the structure of DNA. Okay, so it was posted everywhere. This is in 1953. My mom was born in 1952. So this is not like since the 1600s, like we have with microscopes and bacteria. Like some of the stuff that we've discussed in class, we've been studying for a really long time. DNA is very new. Very new. Okay? So my mom's, 67? And this is where we're at today as far as DNA and forensics and all that. That's happened in the last century. Like during some of you it's your parents, some of you it's your grandparents because you're much younger than I am. But still, what I'm saying is it's not old science, okay? But I will come back to these two gentlemen here shortly. So, I want you to know the timeline, and if you're using the notes that are posted in eCampus, I've kind of rearranged the order and added years because I wanted it to make sense chronologically. Like I wanted to see where we started and how we got there, and so on. So I'm going to begin with this first experiment. This is Griffith's experiment, and it was conducted as far as notes are concerned in 1928. That's when the major findings were reported. But what he did was he gave mice pneumonia. Now, why is it acceptable for him to give pneumonia to mice? What is it about mice? Okay, yes, they're systemically similar to humans. There was a term I taught you earlier in this semester that we use when we reference anything we can test on. What was that term? Model organism, yes. Mice are model organisms, meaning we can test on them. They reproduce really quickly. They are easy to maintain, so easy to feed. And if they die, nobody cares. They're perfect to test on. They're what we reference as a model organism. And I mentioned when I taught you that term, when I say model organisms, people are like, oh, they must be really pretty. No. Like I'm saying, it's ideal to test on these organisms. So that's what I mean by model organisms in science. So he gave these mice pneumonia. And it was okay. No one got upset. Now, pneumonia is caused by a bacteria, and I'm about to ask for Ask for a throwback because I know you know this answer. But the name of this bacteria is Streptococcus pneumoniae. Can you tell me what shape this bacteria is? Spherical shaped. The name tells you the shape. So when you're looking at that type of bacteria, you know that it's going to be, just the name alone tells you that it's going to be spherical or a round shape. Like when you get strep throat, that's Streptococcus. Same thing. So when you have a bacterial infection, a lot of times we can figure out the shape of that by the name of the infection or the name of the bacteria that's infected you. So he gave them strep throat, which is caused by Streptococcus pneumoniae. And there are two strains of strep throat. There is the smooth and the rough. Not strep throat, sorry, pneumonia. I'm trying to mix what I'm teaching you. So the smooth was the S strand. So S was smooth. And that meant that this bacteria had a smooth external capsule. So it kind of looked like that. The R strain, R stood for rough, which means that this bacteria did not have a capsule, so it kind of had a rough external anatomy. And of course, because these are bacteria, they have one circular chromosome. That is not the nucleus, that is their genetic material. Because they don't have a nucleus because they're a bacteria. So the difference between smooth and rough. Now, his experiment. He gave one group of mice the smooth strain of pneumonia. And when he inoculated those mice with the smooth strain of pneumonia, you can tell by this picture what happened to them. What did they do? They died. And how many times do you think he did this before he made the conclusion the smooth kills? A lot. You can't just do it one time. It has to be repeated. So he collected enough data to say, when I inoculate them with the smooth strain, they're going to expire. So recorded that in his notes. So then he said, okay, I'm going to take the other strand, the R strain, the rough strain, and I'm going to inoculate a group of mice with that. And guess what happened when he inoculated those mice? They lived. He was like, okay, what? So it's two types of pneumonia. One has a capsule, one doesn't. The one with the capsule kills. The one without it doesn't kill. And of course, he's repeated this over and over again. And when I say kill, he uses the term virulent. So the smooth strain was considered virulent. The rough strain was considered non-virulent. So he recorded that over and over again and came to the conclusion that smooth kills and rough does not. So then in his inquisitive mind, he thought, I wonder what would happen if I added heat to the smooth strain. So first of all, bacteria, is it living or non-living? Living. And when you add heat to a living organism and you continue to add heat, what eventually happens to that organism? It expires. It dies. So he heated up the smooth strain. And ultimately, what did that cause the bacteria to do? After he heated it up, they expired. They all died. So he did what we call heat killed. So he heat killed the S strain, and he inoculated the mice with it, and guess what happened? They lived. So he took the S, he killed it with heat, he injected, nothing bad happened. Of course, he repeated over and over again, and so now he's not really sure because at this time, DNA is not a thing. They don't know DNA like we know DNA. This is in the 20s. They think that the reason you look like your parents is because of protein. They don't know that DNA is your genetic material. Like, this is part of my story here today. So he has no idea how it is that he could have this kill, heated up, and now it doesn't kill. So he's like, what? So then his thoughts were, what happens if I take the two things that don't kill and I combine them? That's a good question. What happens? You would think this doesn't kill, this doesn't kill. Together, they won't kill. Well, when he did combine them, it killed the mice. And he is completely blown away. Had no way of explaining it. In fact, he said, because, again, that we don't even know about DNA at this point. He says, I'm not sure what happens, but something is transforming in this bacteria. And so we actually call it Griffith's principle of transformation. Because he could explain it. I'm not saying that he was not intelligent, but ultimately, all he could say was something is transforming. He wasn't really sure what was happening, but he knew when he put the two together, something was transforming. So we call it Griffith's principle of transformation, and that's why I kind of highlighted the term transform. Now, that's his experiment right there. He tested on these mice, gave them the smooth and rough strand of pneumonia, and ultimately concluded that when he combines two things, that they transform. I'm going to explain to you what actually happens. This is not something that's part of his experiment. This is me making sure you understand the science behind it. So when he heated up or added heat to the smooth, it killed it. True or false? So all that's here now, you have a dead organism, and it has its shell or its capsule. So it's kind of like an abandoned shell on a beach. What happens to an abandoned shell on a beach? It gets picked up, or some organism that will benefit from it will crawl into it and do what? Live in it. So when this is just an empty shell, what do you think the rough does? What it said was, I really want that shell. I really want that shell. So technically what it did was, this guy's dead. I'm just going to take his DNA and make it part of mine. And now that it's part of mine, I will make a shell. That's what was transforming. But he didn't know what DNA was. Okay, he didn't understand it like that. It would be the same concept. And remember, they're just one cell, so that's very easy for them. But if I was really fast and Claudel could fly, okay, I'm fast and he can fly, but I really want to be fast and fly. So I wait for him to die, and then I get his gene for flying, and I take it in, and I make it part of my DNA, and now I have the ability to do what? Fly. The reason this doesn't work for humans, though, is because we have so many cells. In bacteria, they're just one, right? So it's very easy for them to transform. That's why they mutate so quickly. But for you and I, in order for me to truly be able to fly when he's knocked off, it would have to be in all of my genes, all of my DNA. And that's just pretty much impossible. Okay? But that's what happened there. That's what that transforming actually was. Proceed with your question.
In his experiment, who's the one why he didn't
R strain up?
by himself?
That's a good question. I don't know the answer to that. I do not know the answer to that. Why he didn't heat the R? I wonder if it's because to see what would happen, if it would perhaps become virulent as a result of that. That's a great question. I do not have an answer for you, an honest one at least, that I can give you. So fast forward 10, 15 years. We're in the 40s. We have a group of 3 scientists, Avery, McCarty, and McLeod. They replicate Griffith's experiment. And how are they able to replicate Griffith's experiment exactly like he did? He did it on mice. Tell me how they knew exactly what he did. He wrote it down. He took notes. He said, I did this, and I repeated it so many times. And every good scientist will do that. Every good scientist will do that. So he recorded good, made good notes, made good observations, and these scientists followed it up redid the experiment, got the same results, but their goal was to figure out what is it that is transforming. And after they carried out a series of experiments, they concluded that it was DNA that was transforming. It is still not known that DNA is your hereditary information. They do not know at this point that DNA is passed down. They do not know that. But they do know what it is to an extent. They're just not sure what exactly it does. Okay, which is, again, going to be part of my story. So we had fast-forwarded 10 years, 15 years. We are repeating Griffith's experiment. Avery, McCarty, and McLeod said, hey, it's DNA that's transforming. What did they think your hereditary molecule was? Protein. That's what the world thought at this point. Okay? So that was their experiment. Nothing really new there other than the fact that they said it's DNA that's transforming. Then, jump ahead again, another 10 or so years. And with time, in all things, the technology gets better, the science gets better, the research gets better. So we're learning as we're going through all of this. So that's why it's important for me to put the dates and you kind of see how it all fits together. So here we fast forward a little, a decade more, and we have Alfred Hershey and our first female, Martha Chase. So we call this the Hershey and Chase experiment. And this is happening in 52. And big picture, they're giving bacteria a virus. Can you tell me why it's okay for them to give bacteria a virus? They're a model organism. When that bacteria gets a virus, no one says, oh, what have you done? Nobody cares. Okay? And if you want to kill that bacteria, drop some bleach on it and we're good. We're done. Okay? So bacteria are model organisms and they tested on bacteria. There are a couple things I want to point out. First of all, this word here in blue, bacteriophage. Bacteriophage is the name of a virus that can infect a bacteria. It's the name of a virus that can infect a bacteria. So a bacteriophage is the name of a virus that you give a bacteria. And so what Hershey and Chase were trying to do, they were trying to figure out what molecule is your hereditary molecule. What molecule is passed from parent to offspring? Okay? And to do this, they used viruses. First question, are viruses living or not living? They are not alive. Okay? So our immune system will react to them and it'll kill the cells that are infected with the virus, but our immune system cannot kill the virus. We have to let it run its course. Okay? But every virus you've ever had has tagged you with a little bit of DNA. And that's why you can't get infected with the same virus twice, because your body recognizes it. Okay,
so
proceed.
When they're giving out pneumonia...
No, pneumonia is a bacteria.
That's a bacteria.
Flu shots is, I think, what you're trying to talk about.
Yeah, so I didn't know which one.
Oh, you're good.
Because I was saying, for example, when they do administer the flu shots, and then but you still wind up contracting.
Fantastic question. Let's talk about two reasons. Number one, there are different strains of the flu. One year it's H1N1, H1N2, and these are the names of the viruses. And then it's influenza A, influenza B through F. So the flu viruses change. They mutate. So what they do when they administer flu shots is data has been collected in the previous 6 to 8 months, and they've determined which strand has been most picked up, most popular. And they create a vaccine for that one. So let's say that it's influenza B. So you get your flu shot for influenza B, but you're exposed to A, and guess what? you're getting the flu. The bottom line. That's the first reason, because there's different strains. And because they're viruses, when the virus gets into my cells, if one of my cells happens to mutate, that virus slightly mutates too, if they happen to be together. So then that's how the virus continues to mutate and change. That's why we can't figure out AIDS and HIV, because it mutates so fast. And it actually goes backwards, but that's a whole nother lecture.
Yeah, because I wound up taking a pneumonia shot, but I still caught it.
Yes. So the second reason is you're not supposed to get a flu shot if your immune system's been compromised in any way for the last 7 to 14 days, meaning if you don't feel good. Because if your body is already weak and compromised, and then I expose you to a virus, the likelihood is it's not strong enough to do what? To fight it. So there's different reasons, but that's an excellent question. And people ask that all the time. They're like, I don't even believe in flu shots because every time I get one, I get the flu. I hear that stuff all the time. But I completely agree. I understand it. But the reality is there's so many different versions that, yeah, we're just going with what's most popular that season. Yes, ma'am.
So if you said there's like A through F.
Yeah, like there's all different types of flu. Yes.
So if you get the influenza B, you're not supposed to get it ever again.
Right. You won't get that strain. But you could get C, A. Yeah. So bird flu, whatever. Like, yeah. You can get all of that as long as it's not the same strain that you've already had. So when a virus infects you, it tags you. Okay, so that's where, and those were great questions, so I totally don't mind answering those. So it tags you, so you don't get it again. So when we give these bacteria a virus, and it tags it, it becomes part of whatever molecule will be passed on. Does that make sense? So what they're trying to figure out is which molecule is it that's passed on? Is it DNA? Or is it protein, like we've thought this entire time? So they create a virus, one vat of virus that is all protein-based. So here we have a viral protein, and we make it fluoresce. We make it radioactive so we can trace it. Remember, we use isotopes. That was chapter 2 to do that, okay? So we make it radioactive, and we give it to these bacteria, and so the virus uses the bacteria to make babies. If the protein is what's used and passed on to the kid, then all of these new viruses will all have that protein, that radioactive protein. Guess what happened? We put these viral phages, these viral protein phages, into these bacteria. These bacteria were hijacked, but all the progenies, all the babies, none of them had that radioactive gene. So they realized at that point, and did they just do this one time? No. And this was easy to do multiple times because it's bacteria. So you can have a few vats of bacteria do this and easily see the results. It's not like you have to wait forever. But all of the babies didn't have that. So are proteins passed from parent to offspring? No. On the flip side, they did the same thing. They gave a viral phage that was DNA-based. They hijacked that cell, used it to make progeny or offspring. And I don't want to say offspring because they're not alive. Okay, so I'm going to say progeny. And each of these bacterial phages that was created as a result all had that radioactive phage. So it was Alfred Hershey and Martha Chase that decided DNA is your hereditary molecule. DNA is what's passed from parent to offspring. Okay, so far we have Griffith, who said something transforms. Avery, McCarty, and McLeod came back and said it's DNA that transforms. Hershey and Chase came back right after that and said DNA is your hereditary molecule. Okay, that's where we are so far with the scientists. Now, this is something that I added to review what I've taught you for the first lecture exam. There were 4 biomolecules: carbohydrates, proteins, lipids, and nucleic acids. So nucleic acids is what we're studying right now. DNA is a nucleic acid. I want to remind you that nucleic acids are made up of nucleotides, and nucleotides are the monomers. Do you remember what a monomer is? Just one. Just one. So one piece, the smallest piece that DNA can be, is called a monomer. It's a nucleotide. Each nucleotide, and I've taught this to you already, but has 3 parts. It has a sugar, a phosphate, and then a nitrogen base. There's a sugar, a phosphate, and a nitrogen base. So like right here is one nucleotide. Sugar, phosphate, nitrogen base. Here's another nucleotide. Sugar, phosphate, nitrogen base. So DNA is made up of multiple nucleotides. Also in that chapter, I talked about the bonds you will find. In DNA, there were 2 bonds. It was the only biomolecule that had 2 bonds that we discussed. The first bond was between the phosphate and the sugar, and that's what we call a phosphodiester bond. A phosphodiester bond is the type of bond that you'll find here along the sides. And then the bond that's found here in the middle between the nitrogen bases is called a hydrogen bond. Which one is stronger? Phosphodiester. I don't want you to forget that the weakest of the bonds is the hydrogens. Fantastic. So I just wanted to kind of put that in there real quick, because at this point we know DNA is the hereditary molecule. Okay, we figured that out science-wise. That's where we are in the research. I wanted to remind you of what we know about DNA and the bonding pattern. And now I want to go back 2 years, because it fits in, but I want the years there too, to cover Chargaff's rules. And I've taught you this already as well, but here it is as far as my timeline or chronology is concerned for this chapter. Everywhere there's an A, there will be a T. Everywhere there's a C, there will be a G. So A and T, C and G. This is what we call Chargaff's rules of base pairing that were discovered or determined by Erwin Chargaff in 1950. He says if there's an A present in DNA, its complement is T. And if there's a C, it's G, complementary. So if I have 14 A's, how many T's will I have? 14. That would be kind of like a test question type setup. And just like I mentioned in that previous lecture, another one would be like, say you have 100 bases. And of those, 14 of them were A's. How many G's do you have? So you would know you would have 14 A's and 14 T's. So that would be 28. Then you would subtract that 28 from 100. And then whatever number is left, you would divide by 2. Okay? So that you have, all you need is one number to figure all the rest of them out. All right? This was a concept that I had taught you previously. Another thing I taught you when I taught you these base pairs was that A and G have 2 rings, so they're called purines. T, C, and then it was also U, but we're not on RNA this chapter. But T, C are pyrimidines, which means they have just 1 ring. It means they have just 1 ring. So chronologically speaking, we have Griffith saying that something's transformed. Avery, McCarty, and McLeod saying it's DNA that transforms. Hershey and Chase say that DNA is the hereditary molecule. And within that, Chargaff says everywhere there's an A, there's a T. Everywhere there's a C, there's a G. Okay? Then we have Rosalind Franklin. Rosalind Franklin is one of my favorites. It's really sad how it all worked out for her. I choose to teach her in the extent that I do because I think history is important to why you can answer the test questions, because you remember the story. But Rosalind Franklin was another female in this whole track of DNA. And what she did was she took x-rays of DNA. Lots and lots of x-rays of DNA. And to this point, it has been pretty much thought that we know the structure of DNA, but we think it's just a solid ladder. We just think it's a normal ladder shape with the rungs on it. Well, when she was taking the x-rays of DNA, she saw like a crossing. She said, there's no way it can be just a ladder like this. There has to be some type of turn or cross, because if there wasn't, this wouldn't show up like this. And of course, she had done this multiple times in order to collect data. So her discovery that we credit her for now is that she told us that based on taking an x-ray, that DNA had a helical shape to it. But unfortunately for her, she was working with this radiation machine over and over and over again, day in and day out. And guess what it caused? It caused her to mutate. She got cancer. She got ovarian cancer. And she died at 37. Shortly after, we figured this all out. Okay, so that's sad. And a lot of people don't know about Rosalind Franklin because she never got an award for her findings because she died really young. But look at the year here. 53. She makes her discovery. Then we have Watson and Crick. Same year. Listen to how your book describes it. Watson and Crick brought together information from several sources to work out the DNA structure. They used everybody else's work, put it together. Look what we did. Who got the Nobel Prize? Homegirl was dead by the time they realized that she had actually done. Look at the years. Like, she takes the x-rays, she rolls out, and they're like, and she's not there to say, what? Like, this is me. It's crazy.
Didn't she take her own notes?
We didn't know until after she was dead, for a while, that her exact contribution. But the Nobel Prize cannot be awarded after death. So she was never recognized professionally during life. Her family wasn't recognized because she was gone. And now we talk about her in history books because we realize it. But does everybody teach it? No, not really. Not really. So you can only imagine how that all works out. But Watson and Crick coming out with the, I don't want to say that they're thieves, but I mean, I don't know them personally, so I'm judging based on what, yeah. There could have been a lot more people that could have received some type of like high five.
Did they do any kind of work? Or they just
Great question. There was no actual experiment. Now, they experimented with, like, what does the structure look like? But never actually, as far as this is concerned. Yes. So, just a tidbit of history, right? But you won't forget it, I guarantee it. So, what we know about DNA structure after all of this is combined. We know that DNA is described as 2 polynucleotide chains. What does polynucleotide mean? Many nucleotides. So remember, a nucleotide is a sugar, a phosphate, and a nitrogen base. So many nucleotides. So 2 polynucleotide chains. That makes sense. DNA is 2 polynucleotide chains. And they are twisted around like a double spiral staircase. I will tell you this. You'll hear spiral staircase. You'll hear double helix. You'll hear twisted ladder. It's all the same thing. It's just how they're describing it. Okay? So ultimately, you need to know that it twists. The second descriptor, the 2 chains are anti-parallel. Let's talk about what anti-parallel looks like, because this is your first exposure to it, and then I'm going to come back to it over and over. Parallel means that they run the same. DNA is read 3' to 5'. And I will explain priming in just a moment. Not just a moment, but it'll be a couple slides. But this is how you say this. 3' to 5'. Okay, so it always grows. It always goes in that direction, points in that direction. And I'll also tell you what points means. The second strand is 3' to 5'. So if they were parallel, there would be both 5s at one end and both 3s at one end. But they're not. They're anti-parallel, so they go the opposite way of each other. And I'll explain this as we get in a little bit further. But so this is what we call anti-parallel. Anti-parallel. So DNA is 2 polynucleotide chains. The strands are anti-parallel. We know that everywhere there's an A, there's a T. Everywhere there's a C, there's a G. And if it has a T, you know you're dealing with DNA. Excuse me. If it has a U, which we'll talk about on Thursday, it's RNA. All right? And each full turn, so it's a right-handed turn, and it's described as that because it twists to the right and not to the left. Each full turn from here to here has roughly 10 base pairs. So 10 rungs. Okay? That's basically how we describe the structure of DNA at this point in time. So the structure of DNA. Any questions on the structure of DNA? So takeaway 1, I wanted you to know what the structure of DNA is, or what it is, and how we got to this point. And it's only been in like your parents/grandparents' lifetime that we've discovered all this. Okay? In my lifetime, in the 90s, we figured out what DNA does. So that's crazy. All right? This is a very new science. So,
Are they still working on
oh yes. Oh yes. I don't know exactly what it's I want to say 96. They finally finished. It took them 13 years to take one strand of human DNA and figure out what each gene was. 13 years. It's called the Human Genome Project. 13 years to figure out what our DNA does. That's incredible. And they finished in the 90s. So they had started. And so this is science when it comes to DNA is just one of those things that everybody's trying to jump into. And now it's like stem cells and gene therapy. And we know now gene therapy because we know about DNA and DNA being made of genes. It's crazy. It's just the spot to be, and there's a huge growth opportunity there as well. So that was takeaway 1, structure of DNA. Next is DNA replication. But my first question, of course, I always want to start with a question. Why do we need to replicate DNA? So we can grow. What do you mean by that? Reproduced? Are you talking about, what type of reproduction are you talking about? Okay, gene reproduction? Protein reproduction? I'm going to need more. What'd you say? Why mitosis and meiosis? They're making copies of themselves. So, I need, where's my DNA? Everywhere. In the nucleus of every cell in my body. So when I went to make this cell, I had to copy the DNA so that my new cell would be identical. So why do we replicate DNA? Because we need it as we make these new cells. So that the new cells do exactly what the old cells did. That's why we copy it. When do you and I copy? When does DNA get replicated? Not yet. What'd you say? At all times, there's a spot, and I've already taught this to you. S phase. S phase of interphase. Remember, interphase is G1, S, G2. During S, it's synthesis. What occurs? DNA is replicated. So now you know why we replicate DNA. Now you know when we replicate it. During S phase of interphase. Now I need to teach you how. So we know why and when. Now I need to teach you how. Which brings me to the very last set of scientists that I am going to discuss today. Meselson and Stahl conducted an experiment to figure out how DNA is replicated. And they did this in 1958. So we learned the structure in 53. And by the time 58 came around, we're trying to figure out exactly how is it replicated. So you can tell it's just science. It's like technology, the iPad, like we're just trying to compete with each other to have that next discovery. So what Meselson and Stahl, what they were researching is how is DNA replicated? They had three hypotheses. And I'm going to explain each of these, and I want you to analyze what it is I'm saying and kind of make a decision. Which one do you think is the actual way DNA is replicated? Hypothesis 1, semi-conservative replication. Semi-conservative replication says I have a strand of DNA, a parent strand, right here, and that parent strand separates, and each of these strands serve as a template for a copy to be made, and then when they're done, they're done. So I had one strand, it separated, and served as a template for the two new strands. So at the end, each daughter strand was half old, half new. Semi-conservative. That was hypothesis 1. Conservative is hypothesis 2. Conservative says that I started off with one strand of DNA, the parent strand, and it's separated, and I used each side as a template. Then the copies that were created separate and come together to make a full strand, and then the parent strand goes back together. So I started off with my parent strand, and whenever the process of replication is over, I have a full parent strand still, and this daughter strand that was constructed. Using these, so they were both separated, and then they went back together. That's hypothesis 2, conservative. Hypothesis 3 says, I have my parent strand of DNA, and I'm going to fragment it. I'm going to put it into a lot of small pieces. And each of those small pieces is going to create the other part of it, its complement, and then they're all going to construct themselves back together in order. We call this dispersive replication. Dispersive. So again, hypothesis 1 says I start off with a parent and my product is half old, half new. Hypothesis 2 says I start off with my parent, they replicate, they make their templates, then the templates go back together, so the parent goes back together, and then the new DNA goes together. And hypothesis 3 says I have my parent DNA and it fragments, we cut it up into small pieces, they make their complement and they reconstruct themselves so that the new strands have pieces of old and pieces of new. Which one of these seems to you the most reasonable? If you just had to take a stab in the dark. The B? In science, we have, and this is technically used for evolution and phylogeny, but we have what we call the principle of parsimony, which basically says, whichever explanation is the most simplest is the most likely. Now, I realize that what I'm talking about with that reference is evolution, but when I look at these and I listen to the explanations, the one that's most simplest is probably most likely. Which of these seems like the most simple and straightforward? B? How are A and B different? Which one has an extra step? B has an extra step. Why does it have an extra step? Because I have my parent strand, I created my two templates, and then the templates had to separate, the new strands go together, and the old strands go together. Versus semi-conservative that says, I had my parent strand, I separate, I make my new strands, and I'm done. based on this, okay, so that was the explanation part. They of course tested, I bet you can't guess what they tested on. Mice or bacteria or some model organism. Indeed, they did use bacteria. And they took the genetic material from that bacteria and they made it really, really heavy so they could trace it. So they made it really, really heavy with a heavy nitrogen concentration. Anyways, as they go through this and they run all three processes, of course, over and over and over and over again, so that we can have data to back it up, the one that gives me the consistent outcome every single time is semi-conservative. So based on their research, Meselson and Stahl determined that DNA replication was semi-conservative. DNA replication is semi-conservative. DNA replication is semi-conservative. So this right here is the description for DNA replication, which of course I'm going to teach you. But I have my parent strand. It separates, and each old strand serves as a template to something new. Okay? It's much easier to be successful when you have a guide helping you through step by step. It's less likely that you succeed when things are just kind of all a mess and you have to put it all back together. So this does make the most sense scientifically. So hydrogen bonds will break, the two strands will unwind, each serves as a template. The finished product is half old, half new. Here's the big takeaway from this slide. DNA is synthesized 5' to 3'. DNA is synthesized 5' to 3'. What does synthesized mean? Made, yes. It means that it's made. DNA is synthesized 5' to 3'. All right. So now I have to teach you the process. I'm going to give you a moment to get down these enzymes. If you are pulling off at eCampus, there's a slide with the enzymes on it, but they're not in the right order. So what I've done is I've put them in the correct order, and I've beefed it up by putting the extra steps. So I've numbered them so you can tell what happens where. So I'm going to give you just a moment to reference those, and then I'm going to explain them in my own words. Okay, and you are, of course, are welcome to write as I explain. But first of all, where will I find DNA? In the nucleus of every cell in my body. And the DNA that I find in every single cell is relatively 6.5 feet. 6.5 feet of DNA in every single cell. Okay? So how do you think it's organized in there? Especially when it's going through interphase and all of that. Just like all clumped together. In my head, I think of it, remember that phone cord example I gave you when we were studying proteins? I stretch the phone out here, and then when I come back, it just kind of tangles up, right? That's what I think of. That's what it makes me think of, like it's this massive tangle, because it's a whole bunch in a small, small space. In order to replicate it, though, I have to get it to relax and spread out. Enzyme number 1, topoisomerase. And how do I know that that's an enzyme? It ends in ACE. That ASE indicates to me that this is an enzyme of some sort. Topoisomerase is responsible for loosening that tangle. So I kind of think of it as like a detangler. Okay, it kind of relieves the tension of that DNA molecule. It kind of says, hey, we're about to copy you, so I'm going to need you to kind of relax. Just relax. So topoisomerase is what starts it. Then right behind it is helicase. Helicase is the enzyme that breaks the hydrogen bonds between the two strands. So it goes in and it breaks every single hydrogen bond. I don't know what type of music that you listen to, but I listen to a lot of different musics because I have types of music because I have 4 kids, of course, and then my husband, and then whatever. When we're on a road trip, it's just like a constant. But I listen to lyrics. Like, I want to know what it is they're talking about. So some songs are quite disturbing, and you don't know it because you like the beat, right? Well, we were listening to a rap song, and one of the lyrics is, I'm going to act as helicase and unzip your jeans. And I was like, did he just say helicase? Like, because only smart people would get it. Because helicase is the enzyme that unzips your DNA. And your DNA is referred to as your genes. I was like, like some rappers are super smart. Some are super stupid. And I was like, gee, this guy's smart. He said he's in jail. I said, it doesn't matter. He had a good teacher at some point. If he's going to put helicase in a rap song, that's a thing. Okay? So I was impressed. So that's how I remember helicase, and I always tell people that because it will help you remember, like helicase unzips your genes. Okay, good. So topoisomerase detangles it and kind of loosens it. Helicase unzips it. But because it's been so tangled and wound up, what do you think it naturally wants to do after we separate it? Go right back together. So in order to prevent that, we have our single-stranded binding proteins. Our single-stranded binding proteins will hold it open. Single-stranded binding proteins will hold it open. So it's relieved of tension, it's unzipped, and now it's held open, and it forms what we call the origin of replication. What does origin tell you? Like a beginning, or like where it came from. Like this is a starting point. And I'm going to show you a diagram, of course. I just want to talk you through these terms, because if you know the terms, then it's easier to learn the process. So I established an origin of replication. Once I know where I'm going to start, I have a primer that I put down that will begin this process for me. Serve as a starting spot. And of course, I'll show you that in a diagram. Now down to DNA polymerase. DNA polymerase is actually responsible for adding every A, T, C, G. It actually synthesizes DNA. DNA polymerase synthesizes DNA. DNA polymerase synthesizes DNA. And as we're making DNA, sometimes there's gaps, especially when I explained the lagging strand to you. And because there's gaps, we need something to go back along and fill in those gaps. So the very last enzyme to contribute is DNA ligase. Ligase will come in, and it will fill in all the gaps and seal that new strand. So it will fill in all the gaps and seal that new strand. Okay, so I'm going to show you diagrams and explain as I go. So I'm up here on this first one. I have my strand of DNA that I'm going to replicate. The very first enzyme, topoisomerase, is going ahead of everything, saying, hey, we're about to copy you, so just kind of loosen up. So it's the one that detangles, topoisomerase. It is followed up by helicase that is unzipping, it's breaking those hydrogen bonds, unzipping that DNA. And naturally that DNA, after it's unzipped, wants to immediately go right back, because that's just a natural movement. It's been so tightly coiled these single-stranded binding proteins hold it open. So topoisomerase relaxes, helicase unzips, the single-stranded binding proteins hold it open. As a result, they establish an origin of replication, or in this case right here, it's referred to as the replication fork. They're not exactly the same thing, but they're close enough that you can reference that. So it's kind of like a fork in the road where you can kind of see, and that's where it's replicating. The reason why that fork is created is because replication is taking place. So that's an actual spot when we say the origin of replication or the replication fork. It's a physical spot, but it changes as we continue to go through this process because it will continue to grow in this direction. So those are the first four on the left side if you wrote down those enzymes. Topoisomerase, helicase, the single-stranded binding proteins, and then the origin of replication or the replication fork. When we get to the right side, it talked about an RNA primer. In these RNA primers, you can see, first of all, do you remember what direction DNA is synthesized? 5 to 3. So on this first parent strand, this parent is 3'. The parent is 3' on this first strand, and the 5' end is down here. That works out really well because DNA is anti-parallel. So I put my primer here, and I'm going to point in that direction. I'm synthesizing 5 to 3. So this is the direction that I'm going to go in. That's naturally going to work out really well. This is going to be my leading strand because it's going to happen faster because it's naturally in that direction. But this strand right here is my 5' strand. So the 5' end is here, and the 3' end is there. I can't put a 5' primer here because 5 and 5 can't be together because it's anti-parallel. So in this case, my primer has to be set a little further ahead, and it points backwards. So I'm going to go in this direction as I synthesize. But once I get to the end, I'll have to go put another primer and go back, and another primer and go back. Why do you think we call that the lagging strand? It's slower. It's a slower process. Here is my primer, and this is my leading strand. My primer is set. I'm synthesizing DNA, and here's my DNA polymerase adding the A's, the T's, the C's, and the G's as it's going. But in the lagging strand, I have my primer, and I'm going in this direction. But once I get to the end, I'm going to have to go put another primer and come back. And then once I get to this one, I'm going to have to go put another primer and come back. So this is considered the lagging strand because it stops, it pauses. It's what we call discontinuous. So as it continues, you can see the leading strand is just nice and quick. But the lagging strand, I have my primer, I went backwards. Primer, go backwards. There was a scientist who discovered that this process was occurring slower in the lagging strand. I bet if you look at this diagram, you can figure out that scientist's name. Okazaki. Okazaki is a Japanese scientist who figured out how the lagging strand was being synthesized. And so he wanted you to remember him every time. He calls those fragments Okazaki fragments. So the lagging strand is built by fragments, and those fragments are referred to as Okazaki fragments. So he was able to explain that. It is still DNA polymerase that is adding the A's, the T's, the C's, and the G's. That's what DNA polymerase does. So you can see that this one still on the leading strand is nice and continuous. There's no gaps. But here on the lagging strand, there's a gap. And what enzyme is responsible for sealing it? Ligase, yes. So ligase comes around and it seals that bottom one so that at the end of DNA replication, I have two strands that are half old, half new. Half old, half new. This process is considered semi-conservative. because it's half old, half new. That's my first run through. Now I'm going to go back and explain it again. For DNA to be replicated, first of all, we have to replicate it because each new cell that's created has to have an exact copy of the genetic material as the previous cell. We know that DNA is replicated during S phase of interphase. So we know why and when. This process is how. We know that this process is semi-conservative, which means that my end product will be half old, half new. It begins with the enzyme topoisomerase. Topoisomerase travels along the strand of DNA that's going to be replicated, and it says kind of relax yourself, untangle, so that we can access your genetic material. So we can access these base pairs to copy it. Topoisomerase detangles or relieves the tension. Then it is followed up by helicase. Helicase is the enzyme that comes through and breaks all the hydrogen bonds. So it unzips the DNA. Because it naturally wants to coil right back together, we have to hold it open. And it is held open by single-stranded binding proteins. And as we do this, it creates a point, an origin of replication and a replication fork that kind of travel as we continue to move down the strand of DNA. So this is a physical place, but again, it changes as we are carrying out this process. Now, so I've loosened it, I've separated it, and now I'm ready to replicate it. In order to do that, I am going to need primers. DNA is synthesized 5' to 3'. So for the first parent strand up there, you can see that it is labeled 3'. This is the 3' end. The 5' end is here. That works out well because DNA is anti-parallel. So since I synthesized 5 to 3, I can put my primer there, and the arrow indicates to you in what direction it will go. So it's just going to go this way naturally, and there will be nothing stopping it because it is synthesized in that direction. However, on the lagging strand, the parent strand is the 5' end, 5', and then the 3' here. If I put the same primer right here, it won't be anti-parallel. So I have to do it backwards. Again, why we call it lagging. So I set my primer a little bit ahead, and the arrow indicates the direction in which it will replicate or which it will synthesize. The enzyme that adds the A's, T's, C's, and G's is DNA polymerase, and it adds in this direction. But when it gets to the end, it has to stop. So then it needs to go a little bit further ahead, put another primer, and go back. So that every single time, because DNA can only be made 5 to 3. So every single time, it's going from 5' to 3'. 5' to 3'. This one is already 5 to 3 in a natural direction, which is why we call it the leading strand. And it will be replicated continuously because it won't stop. But the lagging strand is replicated discontinuously because it has to stop, and those segments that it adds are considered Okazaki fragments after the Japanese scientist who discovered that that's how this process was actually being carried out. So I put a primer and then a fragment. Then I'm at the end. Okay, so now I need to put another primer and another fragment. And it's still DNA polymerase in both cases that is synthesizing. That's adding A's, T's, C's, and G's each time. You can see that the leading strand is continuous. There are no gaps. But the lagging strand has gaps, and those gaps are where those primers are going to be. So there would be a primer there. And the enzyme that's responsible for going back and filling in those gaps is DNA ligase. DNA ligase comes and it seals that gap so that it's a continuous strand afterwards. So another primer, Okazaki fragment. Primer, Okazaki fragment. This is DNA replication. What questions do you have on this? I'm about to go through the summary slides, but yes. Uh-huh. This is going from 5 to 3. But on the lagging strand, in order to do that, I can't start right here because it would put 2 5s. So it has to go, it starts here and then it goes back. And once I'm done, I have to go create another primer and grow it back in this direction. Does that make sense? Yes. Yes, ma'am. Yes, ma'am. Go ahead.
So is this one on the board the same as this one? So going from 3 to 5.
Listen to what I said. DNA is synthesized 5 to 3. But when you read DNA, you read it 3 to 5. And that's on a different slide.
Okay.
But that's why I needed you to follow me. That's why I didn't put red here. Because I knew that would be confusing at a point. DNA is synthesized 5 to 3. But when you read DNA, you read it 3 to 5. So here is everything I just said in words. So I pointed and whatever, but here it is in words. I begin with topoisomerase that relaxes the DNA. Helicase unwinds that parental double helix where single-stranded binding proteins will then hold it open. The leading strand is synthesized continuously in the 5 to 3' direction by DNA polymerase. The lagging strand is synthesized discontinuously by DNA polymerase to form Okazaki fragments. After the RNA primer is replaced by DNA, ligase comes in and seals that deal. Claudel, you have a question? No. You're just excited. You're like, yes, DNA replication. So, in my template strand, DNA will always point to the 5', and that's where you got confused. So, let me tell you why we call it 5' and 3'. This is me explaining it. When I taught you nucleotides, I said each nucleotide has a 5-carbon sugar. So what does that mean about that sugar? It's got 5 carbons. Like, that's what it is. So, what we've done is we've numbered the carbons. Carbon 1, carbon 2, carbon 3, and that's a 3 right there. Carbon 4, carbon 5. We've done the same thing. 1, 2, 3, 4, 5. So the 5th carbon, when the 5th carbon is leaving, that's 5'. The 3 carbon is the bottom, is at the bottom of that. So that's the 3' direction. So the tip of this vertex, where that oxygen molecule is, it always points. So in this case, it's pointing this direction. I know that that's the 3' and this is the 5' end. It was just easier for me to teach you on this slide because it had a bigger graphic. So here's my template strand. This is my 3' end. This is my 5' end. When I come in to synthesize DNA, which end do you think I'm going to start at from my template strand? You are exactly right. I'm going to start at this end because DNA is synthesized 5 to 3. So I will add nucleotides. What type of bond is it between the phosphate and the sugar? Phosphodiester. Fantastic. What process allows for a bond to be created? Dehydration. Yes. What type of bond is here in the center? Hydrogen, and it's the weakest of those bonds. So I continue to dehydrate these nucleotides, and eventually I will have my new strand. And look, the oxygens are pointing to the 5' end. So you're always pointing to 5. Always pointing to 5. And that's what I had written on the board. DNA strands are anti-parallel. DNA strands are anti-parallel, so 3 to 5, 3 and 5 here, 3 and 5 here. If they were parallel, it would be 5 and 5 and 3 and 3. Do you remember the shape of our chromosomes? Linear. And how many chromosomes do you and I have? 46, right? So we have a lot of DNA to replicate. So what you and I do as eukaryotes is we have multiple origins of replication, which means we don't start at one end and just work our way to the other end. We start at a lot of midpoints, a lot of origins, and then eventually they all meet up together. Because this process, the product is half old and half new, how do I describe it? Semi-conservative. Semi-conservative. Because my product will be half old and half new, I describe this as semi-conservative replication. DNA is always synthesized 5 to 3. It is only read 3 to 5. The lagging strand will be synthesized discontinuously using Okazaki fragments. The leading strand will be synthesized continuously. DNA is anti-parallel, so the 5 and 3 primes will be opposite one another. Everywhere there's an A, there will be a, everywhere there's a C, there will be a G. DNA synthesis is bidirectional, which means it goes in both directions. And eventually, all of it will meet up. Two more things I have to cover. Quickly, of course, but telomeres. Telomeres are part of, I'm going to draw the structure. I'm not going to tell you the answer. I'm going to ask you. What am I drawing right here? What is this? It is a strand. It's DNA. But whenever I have DNA and it's all wound up like that, how do I refer to it? Chromosome. When it's all wound up really tight, it's considered a chromosome. Okay? And chromosomes are made up of small segments called genes. Right? So you have a lot of genes in your chromosomes. And we'll learn on Thursday that all genes are used, well, the majority of genes are used to make proteins. But at the end of this strand is this box that is not genes. It's what we reference as a telomere. This section is called a telomere. And when a cell is created, and this is another much more in-depth lecture, but when a cell is created, the day it's created, it knows what it will do, what its job is, and it knows when it will die. So they came into this world to create to serve a certain purpose, and as soon as they served it for their extended time, they're out. Like your red blood cells, at max, are in your blood for 120 days before they're gone. And that's maximum. Most of them are 30, 45 days. So, how this cell knows when it's time for it to go. Every single time it replicates, a piece of the telomere is removed. So I kind of think this is like an hourglass type setup. Like as its time passes, the telomere gets shorter and shorter and shorter. And then once it's all gone, what does that cell have to do? It's gone. It's done. That cell has aged. It has reached it's the end of its session. A lysosome will cause it to disintegrate. You'll pee it out. Telomeres kind of give you an idea of how old that cell is. Now some, and this doesn't necessarily apply to all the cells in your body, but I'm going to talk about it. Some of them have the ability, have access to the enzyme telomerase. And this is really more in prokaryotes and cancers, but still. Telomerase is an enzyme. What do you think it does? Telomeres and telomerase. If telomeres are removed as the cell lives, what do you think telomerase does? It literally just adds it back. It's adding or synthesizing those so what does it do to the lifespan of that cell? It extends it. In cancers, cancer cells don't die. Guess what they have a lot of? Telomerase. This is kind of the idea behind anti-aging stuff, okay? But just so you know, this is dealing with DNA. So are you telling me that putting on a topical cream is going to change your DNA? No. Okay? So one of my classes, we had a long discussion about that. We don't have to do all that, but telomerase adds length to that cell or to that chromosome so that it can stay a lot longer. Okay? The telomere itself gives us an idea of how old that cell is. We can use that to gauge how old the cell is. Okay, now what happens if this process goes wrong? What if when I'm replicating the DNA, I make a mistake, which is going to happen? The best thing about DNA is that DNA proofreads itself. DNA polymerase, which is the enzyme that synthesizes DNA, when it realizes it made a mistake, guess what it says? Oh crap, I jacked up. I put a C here and I meant to put something else. Let me get rid of it and put in what belongs. So it corrects itself as it's going. Like that would be like us using autocorrect on our phones. Like it sees that it's done wrong and it immediately fixes it. Now, does autocorrect catch everything? No. No. And sometimes DNA polymerase also misses it. But there is, a lot of times if you're using some type of electronic device, there's a red little squiggly line that shows up underneath it that tells you what? Maybe it's misspelled. Maybe there's something wrong with this word. Well, then what do you do? Yeah, especially like if it depends on who I'm texting. Sometimes I really don't care. But normally I want to sound like I know what I'm talking about. So I'll go back and I'll be like, maybe that's not a word at all. Okay? But DNA polymerase, it goes through and it autocorrects itself, but sometimes it misses words or it doesn't recognize. So it goes back over, and usually most mistakes kind of make a little dent. And it says, oh, like that would be the same as our little squiggly red line. It says, there's something wrong in this area. I can't tell you exactly what it is, and I couldn't correct it, but something's wrong. So you go back, and DNA polymerase says, okay, ooh, I made a mistake here. But instead of removing just the wrong one, it removes all the ones around it. Why? Yeah, I jacked this one up. But I obviously got sidetracked. Or maybe I saw a squirrel or something, right? And I was off for a minute, so I could have messed up a few. So I'm going to go ahead and just take all of you out. I'm going to rewrite that word. Okay? Or I'm going to choose a different word that makes more sense in this situation. So, in the event that something like that happens and they've missed it, it'll come back and it'll fix it. And guess what will seal any gaps that are there? DNA ligase. Polymerase will create it. Ligase will seal it. And all of that is here in the words. Bless you. So yes, there's problems. But our body does a great job of fixing those problems. Here's an example of a very common problem that I want to go ahead and point out because it is on your test. What type of mutation is this? It's what we refer to as the thymine dimer. It's a result of exposure to UV radiation. What normally happens is the A's and T's are placed correctly, but instead of them binding with their complement, the T's bind with each other. What is dimer? What is di? Two. So two thymines just kind of stick together. but it made a little distortion. So guess what polymerase normally does? It comes back and it says, wait, that's a red little squiggly line. And it fixes it. But we have found with time, the more you expose yourself to those environmental assaults, the less effective polymerase is at proofreading. So that's why skin cancer doesn't show up the very first time you get sunburn. It takes a lot of environmental assaults or exposures to sunburns and so on before you actually develop that skin cancer. Okay? So, but this is what it does whenever it's exposed to UV radiation, and polymerase normally comes back and says, hey, wait, you're not supposed to be sticking out, single file line. And you've been underlined with red. Let me come back and fix that. And it will. It'll come back and fix it, and that's why not everybody who gets sunburned has skin cancer. That wasn't a joke.
I did.
Oh. Well, obviously.
Yeah, I know.
All right. The last thing is, first of all, where does this genetic material belong? Whose genetic material is this? Whose genetic material is this? How do you know this is a bacterial genetic? It's a circle. It's a circle. What is a eukaryote? It's a linear chromosome, right? They're in lines, and they're wrapped around. But in a prokaryote or a bacteria, they have a circular chromosome. And when I say a circular chromosome, how many do they have? And what I'm trying to point out here is DNA replication does the exact same thing. But why do I only need one origin of replication? Yeah, I've got one chromosome. It's a little circle. But the process still carried out the same way, half old, half new. That means it is semi-conservative. Meselson installed 1958. This is the end of chapter 14. What questions do you have? Can I re-lecture it?
Yeah.
Absolutely.
Not the whole thing.
Yeah, that's what I'm saying. Absolutely. Yeah. Whatever, Mr. Brooks.